All right, well, here we are, Lecture 20, Chunk B, last chunk of the course. We've already talked about the light reactions briefly, just to get us on the same page, and remember that we've got some NADPH and ATP ready to go here. And here we'll talk about the Calvin cycle in all its glory, the actual biochemical fixing of carbon dioxide into glucose molecules. This is the Calvin cycle. And we'll also talk about, I don't know why it's not blue there, but we'll also wrap up this chunk by talking about this C4 pathway that exists in tropical plants where we can concentrate carbon dioxide so that it's not lost or leached out by these plants that have to live in very hot climates. So here's where we left off in the last lecture, in the last chunk. We talked about where this was occurring, the dark reactions. It's going to be in the stroma. And we set the stage by saying we've got tons of ATP on hand that was made through photophosphorylation and tons of NADPH as well. And I even include you in on the overall net reaction of the Calvin cycle, which is going to involve 6 carbon dioxides, 12 NADPHs, and 18 ATPs. And all of those electrons and all of that energy is going to make for us one single measly little molecule of glucose that we can eventually, as a plant, go off and store as starch or use to make the cellulose of the plant cell walls. That's what we'll do with that glucose that we make. So let's talk about the nitty-gritty of the Calvin cycle now. In order to do all that we're trying to do, in order to make this glucose, we need to take the carbon dioxide that we're going to absorb and add it to something. We have to carboxylate something. We have to tack that carbon dioxide molecule onto something the plant already has. And the molecule we're going to use for that carboxylation is ribulose 1,5-bisphosphate. Now, the 1,5-bisphosphate will be familiar to you, of course. We've dealt with other bisphosphates this semester. Ribulose is the only new player to us, and ribulose is a 5-carbon sugar. So we're going to absorb 6 different carbon dioxide molecules and add them each one at a time to 6 different ribulose 1,5-bisphosphates. And we're going to turn 5-carbon ribulose 1,5-bisphosphate into a 6-carbon molecule. So that's going to make 6 different intermediates of this 6-carbon molecule, and this intermediate is very, very unstable. And it spontaneously collapses, it spontaneously hydrolyzes into 3-phosphoglycerate. So, if we had 6 ribulose 1,5-bisphosphates, and we added to them 6 carbon dioxides, we had 6 unstable intermediates that collapsed down into 2 molecules of 3-phosphoglycerate each. That means we have a total of 12 molecules of 3-PG when this is all done. Our 6 unstable molecules broke down into 12 3-PGs, each of them 3 carbons each. And so that's what we show here. We have ribulose 1,5-bisphosphate that is carboxylated and instantly breaks down into 2 3-PGs each. If we have 6 ribulose 1,5-bisphosphates, we've just yielded 12 3-PGs. And yeah, this is 3-PG. This is an old buddy of ours. Of course, we've seen 3-PG plenty of times, both in glycolysis and in gluconeogenesis. It is indeed the same molecule, a 3-carbon sugar, a triose. Now, of these 12 molecules of 3-PG that we have made, 2 are going to go off and make glucose, a 6-carbon sugar. The other 10 remaining 3-PGs are going to be used to regenerate the 6 molecules of ribulose 1,5-bisphosphate that we just started with a moment ago. So from a big picture, zoomed out, detail-free perspective, this is really all the Calvin cycle does. It takes 6 ribulose 1,5-bisphosphates, carboxylates all 6 of them with 6 carbon dioxides each. Those break down to 12 3-PGs, 2 of which go off and make glucose, and the other 10 are used to regenerate the 6 ribulose 1,5-bisphosphates we started with. This is the roadmap of the Calvin cycle from your textbook. We'll return to this again and again. This is going to carry us through everything we need to know, but I will tell you right off what you do not need to know are the enzyme names or the intermediate names, except for the ones that I illustrate here in this lecture chunk. So anything that I don't specifically refer to here, you do not need to know. We're after the general concepts of the Calvin cycle, what went into that cycle, and what comes out. So let's talk about the sugar first. Let's make glucose before we go any further. As we've just discussed, the first reaction of the Calvin cycle is a condensation reaction. We're going to make a carbon-carbon bond here, and that bond is going to occur between the carbon of carbon dioxide and carbon-2 of ribulose 1,5-bisphosphate. That's going to create this 6-carbon beast, which we will never name, and it's that molecule that's going to break down into 2 molecules of 3-phosphoglycerate. So there's the carbon dioxide joining on with ribulose, and that instantly hydrolyzes because of its instability, giving us our 3PGs. This reaction is catalyzed, this condensation and cleavage reaction, the hydrolysis as well, is catalyzed by a single enzyme named ribulose 1,5-bisphosphate carboxylase oxygenase. Boy, is that a mouthful. But the way that I'm going to ask you to know this enzyme name is the same way that everybody knows this enzyme name in plant biology. We will refer to this enzyme by its abbreviated name, Rubisco. Rubisco is the most plentiful protein in all of nature. There is more Rubisco on this earth than any other single protein, and it is solely responsible for kicking off the Calvin cycle, the dark reactions of green plants. Rubisco is found in the stroma because that's where these reactions are occurring, but it is associated with the thylakoid membrane through a hydrophobic region. And for any of you who've ever done plant molecular biology, or if you know anybody who's done plant molecular biology, especially plant biochemistry, just ask them about Rubisco and you're likely to get a groan. Plants make so much Rubisco that you just can't get rid of it. No matter what protein work you try to do with plant cells, Rubisco contaminates your proteins because it's just so plentiful. So let's see what happens with these 3PGs. Let's make some glucose. Well, these 3PGs are holding on to the carbon dioxide we just incorporated. That was the purpose of the first step of the Calvin cycle. This was the actual carbon fixation step. What's going to happen next is that 3PG is going to be phosphorylated and reduced to create GAP, glyceraldehyde 3-phosphate. That's going to take 2 different steps because it's 2 different processes, and we'll go through a 1,3-BPG intermediate. So all of this should seem very, very familiar to you. It's very much like gluconeogenesis. It's very much like glycolysis in reverse. And you might also note that there in step 3, we just used one of our super-duper NADPH super reducers. In order to convert 1,3-BPG into glyceraldehyde 3-phosphate, we needed to call upon NADPH to catalyze that reaction. Once GAP is made, it can go off and be used to do one of 2 very different and important things for the plant. It can be used to make glucose, or it can be used to help regenerate ribulose 1,5-bisphosphate. We will follow the glucose pathway first, and the rest of it will be very familiar to you. You'll have seen these reactions before. You can even see them here on the diagram. GAP comes together with an isomerized version of GAP that we refer to as DHAP. One GAP molecule and one DHAP molecule are condensed together by an enzyme aldolase to create fructose 1,6-bisphosphate, which is then dephosphorylated to create fructose 6-phosphate, isomerized to glucose 6-phosphate, and then that last phosphate is removed to make glucose. You don't have to learn that. That's gluconeogenesis. You know that. Or you can just remember it as glycolysis in reverse. We've done this already. These reactions are familiar. So that's pretty much all you need to know about making glucose from the dark reactions. Carbon dioxide condenses with ribulose 1,5-bisphosphate. 3PG is yielded from that. 2 3PGs are used to go through a very gluconeogenesis-like pathway to ultimately make GAP and DHAP, fructose 1,6-bisphosphate, fructose 6-phosphate, glucose 6-phosphate, and glucose. So the rest of this chunk, and really what we have to concern ourselves with the most, is how is the Calvin cycle a cycle? If we started with ribulose 1,5-bisphosphate, how do we get back to it? And let's be a little bit more specific. If we started with 6 separate molecules of ribulose 1,5-bisphosphate, how do we get back to 6 molecules of ribulose 1,5-bis again? So in general, regenerating ribulose 1,5-bisphosphate is divided into 4 phases. There's a preparation phase, a reshuffling phase, isomerization, and finally a phosphorylation reaction. Preparation is very much what it sounds like. It's a series of reactions and steps that make all of the different parts or molecules we will need for this regeneration. So preparation begins with taking some of the GAP and a DHAP that we can make from 3PG and using that to condense together via the action of aldolase to create fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is then used to make fructose 6-phosphate. And of course, these are the reactions that also go off to make glucose, but we're going to stop right there, and we're going to make fructose 6-phosphate and leave it at that. Now, another thing I want to point out is we are not doing this to all of our molecules. In other words, we don't take all of our remaining 3PGs and convert them into GAP and DHAP and use those to make fructose 1,6-bis and all of that. No, instead what we're doing is we're making these molecules and we're reserving some. So of the 3PGs that are remaining after we make glucose, some of those 3PGs go all the way back to making fructose 6-phosphate, but not all of them. Others of those 3PGs are used to make GAP and DHAP, and that's it, and they stop at that. So we're making individual molecules here with the remaining 3PGs, and we can see some of them show themselves here in this leg or track of the Calvin cycle. So yes, we take our 3PGs and we make GAP and DHAP, but then we take some of those GAPs and DHAPs, not all of them, and make fructose 1,6-bisphosphate. And then we take some of that fructose 1,6-bis and use it to make fructose 6-phosphate. We're reserving molecules as well. And we're going to use these molecules, GAP, DHAP, and fructose 6-phosphate, as our ingredients for reshuffling. So most of the reactions that are involved in reshuffling of the Calvin cycle are reactions we would have covered had we gone over the pentose phosphate pathway. But we didn't go over the pentose phosphate pathway, so these reactions will largely be new to us. Essentially what happens in reshuffling is what the term reshuffling is meant to capture. These molecules are completely reshuffled into new molecules. This is different from isomerization. In isomerization, you typically take a group or two and rearrange them. The backbone of the molecule typically stays put in isomerization. Reshuffling is far more extensive. A reshuffling reaction is a complete restructuring of the entire molecule. And since we have all of these different ingredients, GAP, DHAP, and fructose 6-phosphate, when we say we're reshuffling those three, we literally mean we're breaking all three of those molecules down into their constituent parts and bringing them together into brand new molecules. So these ingredients will be blown apart almost to their individual atoms and then brought together to make these strange molecules along the left-hand side that we haven't yet talked about in this course. Molecules like erythrose 4-phosphate and cetapulose 7-phosphate. I will not be asking you the names of these molecules on any assessment in the future, but these are the things that we make through the reshuffling of GAP, DHAP, and fructose 6-phosphate. What we will eventually do is isomerize these intermediates into two molecules, xylulose 5-phosphate and ribose 5-phosphate. These molecules I do want you to know because we're near the end of the Calvin cycle and we're about to enter regeneration. So for our purposes, we will call the two products of reshuffling ribose 5-phosphate and xylulose 5-phosphate. Those are the molecules that we will say that reshuffling gives us. And again, when we say reshuffling, we mean the blowing apart of GAP, DHAP, and fructose 6-phosphate in order to rebuild from scratch ribose 5-phosphate and xylulose 5-phosphate. Now, we're going to take both of these molecules, ribose and xylulose, and we're going to isomerize them into ribulose 5-phosphate. That's the isomerization phase or the isomerization step of the Calvin cycle. Now remember, you don't need to know any of the intermediate names. I just want you to know ribose 5-phosphate, xylulose 5-phosphate, and ribulose 5-phosphate. So we're taking these two intermediates and isomerizing it into this. And for those of you who have been tuned in and paying attention, you probably realize that two things are converging here. If we've already made ribulose 5-phosphate, we're one step away from the regeneration that we need in order to make ribulose 1,5-bis phosphate. And you also may remember that the last phase of the Calvin cycle is phosphorylation. And so all we need to do now is phosphorylate ribulose 5-phosphate into ribulose 1,5-bis, and we have regenerated our intermediates. We've returned the Calvin cycle to the beginning. This phosphorylation is ATP dependent, and of course that ATP is coming from the photophosphorylation that occurred in the light reactions. Now everything we just discussed, that is preparation, reshuffling, isomerization, and phosphorylation, all of that was simply to regenerate the 6 ribulose 1,5-bis phosphates that we started with when we absorbed 6 carbon dioxides at the very beginning of the Calvin cycle. It was the glucose arm, that very quick glucose detour, where we took 2 3-PGs and used them to generate a single molecule of glucose. That's what the Calvin cycle is all about. So I'd like to go back and do the Calvin cycle again, but I would like to do it from the perspective of carbons. I want you to really see what's going on here in this reaction by following the carbons. So at the very beginning of the Calvin cycle, we have 6 ribulose 1,5-bis phosphates that are each carboxylated by 1 molecule of carbon dioxide each. That gives us a 6-carbon intermediate, 6 of them in fact, and each of them split into 12 trioses, which are 3-phosphoglycerate. So we go from 6 ribulose 1,5-bis phosphates, absorbing an additional carbon each, splitting up into 12 3-PGs. 2 of those 12 3-PGs go off and make glucose through a process that's very much like gluconeogenesis. Well, if 3-PGs are 3 carbons long and we stole 2 of them, that gave us a total of 6 carbons, and that's great because glucose is a 6-carbon sugar, and so those 3 carbons from each of those 3-PGs are used to build 6-carbon glucose. So the 6 carbons in those 3-PGs are accounted for in 1 molecule of glucose. That left us with 10 molecules of 3-phosphoglycerate that we have yet to use. If each of those 3-phosphoglycerates is 3 carbons in length, we actually have 30 carbons total to play with. Well, what do we do with them? Well, first we take some of those 3-PGs and we make fructose 6-phosphate. And we take others and we make DAP, and we take others and we make GAP, and we prepare all of these different ingredients, but still among all those molecules, all we have are the 30 carbons we were playing with. And then we reshuffle them into these strange intermediates, and we end up with ribose 5-phosphate and xylulose 5-phosphate, but still, in all of that, we only have 30 carbons accounted for. And then we isomerize those molecules both into ribulose 5-phosphate. And still, we have only those 30 carbons, and we phosphorylate ribulose 5-phosphate into ribulose 1,5-bis phosphate. And how many did we make? We made 6 of them. We regenerate the same 6 ribulose 1,5-bis phosphates that we started with at the end of the Calvin cycle. But ribulose is a 5-carbon sugar, and we made 6 of them. So how many carbons are we accounting for? All 30. So we start with 30 carbons. We add a carbon dioxide to each of them, giving us 36 carbons. And we steal 6 of those 36 to make 1 glucose. And the other 30 we use to regenerate ribulose 1,5-bis phosphate and come back to where we started. Every single metabolic cycle must regenerate back to its beginning to be a cycle. And this regeneration here is the most important and critical part of the Calvin cycle. Let's consider the dark reactions from an energetic perspective. The delta G for all of the reactions that create glucose in the Calvin cycle is, my goodness, a positive 478 kilojoules per mole. That's a lot of energy that goes into making glucose. The energy of light itself can be measured per unit mole, and at a wavelength of 600 nanometers, the wavelength that's absorbed by photosystem 2, that energy is of almost 1600 kilojoules per mole. Think about that. The plant is absorbing 1600 kilojoules per mole. and it's capturing, through glucose biosynthesis, 478 of those kilojoules. The book says, therefore, based on this math, photosynthesis is only a little bit more than 30% efficient. And I'm like, what? Only? Yeah, sure, it's true that plants lose about 70% of the energy contained in sunlight when they do the dark reactions, but let's not lose sight of the fact that plants are gathering sunlight energy and using it chemically to make glucose. Like, is that not insane? That plants are absorbing sunlight and making sugar from it? I think it's amazing that something exists on this planet that can fix sunlight energy at all. Never mind use that sunlight energy to make something as organic and complex as glucose. So that's, to me, truly awesome in the truest sense of the word. Okay, so as promised here, let's talk about fixing carbon dioxide in tropical plants. Tropical plants, as I've already told you, have this carbon-4 or 4-carbon pathway, C4 pathway, and this is for concentrating carbon dioxide. The reason why it's called a C4 pathway is because it involves 4 carbon molecules. In fact, it involves 4 carbon molecules that we're very familiar with. Eventually, once we concentrate this carbon dioxide, that carbon dioxide will be offloaded to ribulose 1,5-bisphosphate and go into a Calvin-like pathway to make 3-phosphoglycerates, and all of that will be the same as we've seen before. So all we're going to focus on here is just getting that carbon dioxide concentrated deeper in the cell. So for this C4 pathway, the very first thing that happens to carbon dioxide once it's absorbed by a leaf is that it reacts with PEP, our old friend phosphoenol pyruvate, and when PEP is carboxylated, we get oxaloacetate. Pretty crazy, right? And then from there, it starts to look like the citric acid cycle, but it starts to look like the citric acid cycle in reverse for a step or two, because the oxaloacetate that we make is oxidized to malate, and there's NADPH doing that oxidation. And that malate is then transported deeper into the cell, and it is eventually decarboxylated to make pyruvate. Pyruvate, of course, we're familiar with as well. But don't lose sight of what just happened there. We take in carbon dioxide in a leaf cell, we carboxylate PEP to make oxaloacetate. Oxaloacetate is reduced to become malate. Malate is brought deeper into the cell and decarboxylated into pyruvate. So this is the offloading and the concentrating of carbon dioxide that the C4 pathway exists for. Once malate is decarboxylated into pyruvate, it's this carbon dioxide that the plant wanted all along. And this carbon dioxide can then enter this Calvin cycle deeper in the plant, and it won't be lost due to the high heat that the plant is experiencing. Pyruvate is then phosphorylated back into PEP, and that PEP is ready to accept the next cargo of carbon dioxide and become oxaloacetate once again. So this C4 pathway is actually a C4 cycle. It goes from PEP to oxaloacetate. Oxaloacetate is essentially a carbon dioxide carrier in this case. Oxaloacetate is reduced to malate, malate transported deeper into the cell. Malate offloads that carbon dioxide so that it can enter the Calvin cycle. That yields pyruvate. That pyruvate goes back out to the leaf cell where it is modified to phosphoenol pyruvate, and once we regenerate phosphoenol pyruvate, we're ready to repeat this cycle again and take up another carbon dioxide. Are we burning some ATP to do this? Yes, we are. Are we burning a lot of high-power electron acceptors, these NADPH superreducers? Yes, we are. But remember what makes those for us. We make ATP and NADPH by the action of the sun. Photophosphorylation, as well as the reaction center's reduction powers with NADP, that's where we get these two from. And I'll tell you one thing, there's nothing more plentiful around tropical plants than sunlight. So the tropical plants that are doing this don't worry so much about burning ATP and NADPH for this C4 pathway. They have tons of ATP and NADPH. What they would like is to store excess glucose for later on, and they would also like to have that glucose to build new cell walls. So this energy expenditure, this energy investment is worth it in order to convert carbon dioxide into glucose. And again, big picture, all of this is done to concentrate carbon dioxide, which in very hot climates where tropical plants are living is very easily leached out of and lost by plants, especially in those surface leaf cells. So, to summarize this chunk, the point of the dark reactions is not to make glucose for food, but to make glucose that can be stored as starch or as cellulose in the cell wall. For the carbon cycle, we take 6 individual molecules of ribulose 1,5-bisphosphate and we carboxylate them each with 6 carbon dioxides. That yields a very unstable 6-carbon molecule, but we have 6 of those. Each one splits in half to give a total of 12 molecules of 3PG. 2 of those 12 molecules are used to make glucose, where 3PG is changed into DAP and GAP, DHAP and GAP. The DHAP and GAP are condensed together to make fructose 1,6-bisphosphate, and then we follow a gluconeogenesis pathway all the way to glucose. The other 10 3PGs go through this fairly complicated set of phases that are called preparation, where some of them are converted into fructose 6-phosphate, some of them are converted into DAP. Why do I keep saying DAP? Some of them are converted into DHAP, some of them are converted into GAP, and then we go on to reshuffling, where those molecules are then brought together to create 2 different intermediates, xylulose 5-phosphate and ribose 5-phosphate. Each of those are then isomerized to ribulose 5-phosphate alone, and finally ribulose 5-phosphate is phosphorylated through the action of ATP to regenerate ribulose 1,5-bisphosphate. Once we've regenerated those 6 molecules of ribulose 1,5-bisphosphate, we're ready to repeat this cycle again and do another round of the Calvin cycle, make more glucose. We ended the chunk by saying that these tropical plants have a C4 pathway where PEP can be carboxylated to oxaloacetate, which can then be reduced into malate. Malate carries that carbon dioxide deeper into the cell, into the plant, where it offloads that carbon dioxide to a Calvin cycle, and then back out to the surface. That malate goes as pyruvate and then PEP. And this works to concentrate carbon dioxide deeper in the plant where it won't be lost. But by way of summary, let's back up a little bit more and just make sure that we understand all that we've talked about here. So in a plant cell, coating the thylakoid membrane are antenna chlorophyll. And when light hits that antenna chlorophyll, that light energy excites the electrons of the antenna chlorophyll molecules. Those excited electrons then hop from antenna chlorophyll to antenna chlorophyll, following a random path until eventually, sooner or later, they collide with a reaction center, a specialized area on the thylakoid membrane where we have photosystems. Photosystem 1 takes that excited light energy and uses it to fully oxidize water into oxygen, which is released, but also protons and electrons. The protons are used to pump and create a proton gradient, and the electrons are passed from photosystem 2 to photosystem 1, where they are fully charged and used to reduce NADP to NADPH. The protons are used to flow through an ATP synthase-like enzyme that cranks out tons of ATP via a process of loose, tight, and open conformations. And then we have plenty of NADPH and ATP on hand that we can use for the dark reactions. And in the dark reactions, carbon dioxide that is absorbed by the plants is condensed with ribulose 1,5-bisphosphate. That spontaneously breaks down into 2 molecules of 3PG. But since we're doing this to 6 different ribulose 1,5-bises, we actually get 12 3PGs. 2 of those 12 we can feed into a gluconeogenesis-like pathway that ultimately results in glucose. And the other 10 we use for preparation, where we make DHAP, GAP, and fructose 6-phosphate. Reshuffling, where we change those into xylulose 5-phosphate and ribose 5-phosphate. Isomerization, where we isomerize each of those intermediates into ribulose 5-phosphate, and then phosphorylation, where we regenerate that into 6 new molecules of ribulose 1,5-bisphosphate. But it's the sugar we were after. And those plants take that sugar and they use it to store that glucose for harder times later in the future. And plants store that glucose as amylose and amylopectin. But let's say those hard times came. Let's say there is that cloudy day and there's no sun to fuel the light reactions. What happens to the glucose in starch? Well, that glucose is cleaved and released from starch, very much like it's cleaved and released from glycogen. And that released glucose is then fed into the plant version of glycolysis, where it is phosphorylated into glucose 6-phosphate, isomerized into fructose 6-phosphate, phosphorylated again into fructose 1,6-bisphosphate, cleaved into DAP and DHAP. That DHAP is isomerized into GAP, and now in parallel, 2 molecules of GAP become 1,3-BPG, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenolpyruvate, until ultimately we make 2 pyruvates. Why? Well, we make 2 pyruvates so that we can decarboxylate pyruvate into hydroxyethyl and link that hydroxyethyl onto TPP. We then exchange TPP with lipoic acid because lipoic acid can steal protons and electrons from hydroxyethyl, converting it into acetyl. CoA swoops in and steals that acetyl, yielding acetyl-CoA. And FAD and then NAD consecutively take the electrons and protons off of lipoic acid and eventually shuttle those electrons to the electron transport chain. Acetyl-CoA goes to the citric acid cycle, where the acetyl group complexes with oxaloacetate, yielding citrate. Citrate is isomerized into isocitrate. Isocitrate is decarboxylated. Isocitrate is oxidatively decarboxylated into alpha-ketoglutarate. Carbon dioxide is released as waste, but more importantly, NADH is reduced. Alpha-ketoglutarate then has a second round of oxidative decarboxylation, where another carbon is released as carbon dioxide, and another NAD plus is reduced to NADH plus H. That yields succinyl-CoA. Succinyl-CoA is cleaved to make succinate, the energy of that CoA bond used to make GTP, which then fuels the phosphorylation of ATP. Succinate is oxidized to fumarate. That's succinate dehydrogenase, by the way, complex 2 of the electron transport chain. Fumarate is hydrated to malate. Malate is oxidized to oxaloacetate, and that brings us back to the beginning of the citric acid cycle. But all those electrons have a big role to play because they're going to be moved from complex to complex of the electron transport chain. Electrons are going to arrive at complex 1, riding the NADH shuttle, and they'll be offloaded to FMN, from FMN to iron-sulfur clusters, and from iron-sulfur clusters to our CoQ electron carrier. Protons will be pumped along the way into the intermitochondrial space. CoQ will carry those electrons to complex 3, where they'll cycle through a number of cytochromes until they end up on cytochrome C, which brings them to complex 4. And complex 4 will ultimately put those electrons onto oxygen, yielding water. Succinate dehydrogenase, which played a role for us there in the citric acid cycle, will offload its electrons directly to cytochrome Q, to CoQ, and CoQ will carry those to complex 3 and then to complex 4 and water, as just described. The net result of all of this is a proton gradient across the inner mitochondrial membrane. And plants will do the same thing we do. They'll allow those protons to flow through ATP synthase, spinning the stalk of the F0 region as they do so, and as that stalk spins, it will perturb and drive conformational changes in the F1 region of ATP synthase, causing ATP synthase to cycle from loose, where ADP and inorganic phosphate are bound, to tight, where they are squeezed into ATP, to open, where that ATP is released. And no matter how cloudy that day might be, that plant will survive, living off of the ATP that it can make in this fashion, the ATP that it makes in its mitochondria, exactly the same as the ATP we make in our own. So that, my friends, is undergraduate biochemistry. As I've said all along, and as I will say for the last time here, thanks so much for watching.